So when it comes to actually extracting them, all you do is you open it up, you go to the plant that you're going to be extracting from, and you sort of just like insert the leaf into it, like that. And then when you have like enough in it, you close the lid onto the leaf to kind of tear it off. One thing you don't want to do is you don't want to use a flag leaf. A flag leaf is basically like this guy. You can see how it's like attached directly to the panicle of the seeds. This leaf is responsible entirely for the nutrition that these seeds get. So you never want to take from the flag leaf. You always want to take from like a different leaf further down, like this one or something like that. So with the glass beads, in this case, it's 3 each, so in this case, it's just gonna be 6. You don't want to reach into the container itself, even with the sterile tweezers. So you just shake like a small number like onto the lid, and then you can grab it from the lid. So when you open this up, you want to be careful not to let your tweezers touch anything else, even like the leaves. You sort of just reach in, try not to touch any but the one you're going for, and then just drop it in. And then 2, and then 3. And then you close the lid up, and there you go. It is like the lid, right? You can you unscrew it in order to move everything around. What you want to do is you want to take these, make sure the lid is on very, very tightly, because they like opening up in the homogenizer, and if it opens up, you are very sad. You just stick them in there, and I'll do yours too, just so I can show you everything. Again, make sure it's on very, very tightly. You always want to have it balanced, so you're going to put it directly across. That's not right here. Yeah. And then you can take this guy, just sort of twist it up, so it's covering the top of these. So like it's right here, you sort of twist it up to the right, and then take this guy, screw it back on tightly, and close it up. And then you're gonna run it 2 to 3 times, just until like when you look inside there, it's all powdery. Each time after done running it, you're gonna wanna open this up and then like just make sure the lids are on tightly again, like in between each run. So yeah. So basically, when you're doing your samples, you add 500 microliters of Edwards buffer first. So you're gonna take, take Edwards buffer, make sure it looks about right, and then put it in. And we only have 1 sample right now, so it's not that as big of a deal, but generally you have a lot of samples, so you want to hold it above so that way it doesn't touch anything, because then you can put it back in there and grab more without wasting pipette tips. And you close, you take this, go over to here where we have our vortexer, and then you hold it in the vortexer for like 10 seconds or so. So with phenylchloroform IA, it's the same thing, 500 microliters. Reach in, grab it. This one sort of likes to slip out, so like as soon as you're done taking it, you want to like immediately put it in, because it'll like make droplets everywhere. And in the same deal, if you're doing lots of samples, you want to try to hold it above instead of like in the thing, because you want to be able to not have to waste a bunch of pipette tips. So yeah, and then take this, bring it back to here again, vortex for like 10 seconds. You want to make sure it's all like green and foamy throughout without any separation by the time you're done vortexing. So we have 2 centrifuges. I like this one better because the lid doesn't give me as much troubles. So you turn it on, wait for it to kind of like set up. As soon as it opens, you screw to the left in order to take off the lid. Put these in here. You want to have them balanced. So again, like alternate. Put the lid back on. Screw it tightly until there's like resistance. And then you close the lid here. We're doing it, we want to make sure that the top speed, we're doing it for 5 minutes. Yeah, so it goes up at 30-second intervals. You want to make sure it's 5 minutes, and you just press start. So the 1.5 milliliter tubes are right here. You can usually tell by because they're the colorful ones. They also are labeled, so like 1.5 right here. In order to get the 1.5 tubes, these have all been like sterilized, so you don't want to reach in. Instead, what you're going to do is you're going to kind of lift the corner of it a bit, and then just gently shake them out until you have a number that you want. We got lucky enough that we got exactly 2. Sometimes you go over. When you go over, if you have like 3, for example, what you do is you take any extras, close them up, and then just place them here, like where that one is. So that way people can use them later on. And so then what we're gonna do is we need to label them. So we're gonna label them, mine was 316. We're gonna label the plant number on the top, and on the side, we're gonna say what type of plant it was and the plant number. So in my case, 3KO-2T3, and then 316. So for these ones, I typically use the P200. You can technically also use like the P1000 because it's 200 microliters, but I like using the P200 more because later on you're gonna have a 150 and you're also gonna have a 900, so it's just more convenient, not have to do as much. So this one you're gonna do is you have your layer separation. So you're gonna be taking 200 microliters of the top layer, the sort of like clearish one, and putting them into the new tube. You don't want to take any of the plant material because that could interfere with like the process of DNA extraction later on. So I usually do is I sort of like tilt it a little bit, and then I reach in, careful not to touch the walls because there's often plants there, and then just kind of like take up very slowly. Sort of look at it, make sure there isn't anything in there, like as in plant stuff, and then I can transfer it in. For this one, you are going to want to use new tips between each one because obviously you're putting it in there. You don't want to cross-contaminate. So next step is adding the 150 microliters of isopropanol. Again, you want to use the P200 for this one. For this one, like I said, when you have a lot of samples, you want to just hold it above because that way you don't have to like waste pipette tips. So you put it in here, and then what you're gonna do is you're basically gonna like invert it a couple of times so that just really mixes it like well with everything. This is gonna precipitate out the DNA. So you can't really tell very well on the camera, but like it's a little bit more foggy because the DNA has been kind of pulled out of solution by the isopropanol. Especially when you're going to have it in for 10 minutes, what's going to happen is it's going to cause a DNA pellet to form in the corner. So you really want to make sure that these are all like facing inwards because then it's easier to keep track of where the DNA pellet is going to be. For previous steps, it didn't matter as much, but for this one, you want to make sure that they're all like facing inwards for that reason. So, yeah. All right, so while they're in there for 10 minutes is about the time I want to turn the incubator if you haven't already. So it's gonna be this bottom incubator. You're just gonna flip the switch. Over here, it should be at 37, but just to be sure, you wanna make sure this lines up with that little line where it says 37. And then it's just gonna be heating up while we're doing the rest of it. All right, so basically after you centrifuge it for 10 minutes, you wanna make sure you have the DNA pellet. So again, I don't know if you'll be able to see it in the camera, but basically I use my phone flashlight to look, and you just kind of want to see if at the bottom there's like a sort of a white smudge or pellet that indicates the DNA is there. So once you're certain that you have the DNA pellet, the next part is going to be basically washing it with the ethanol. And first what you're going to do is you're actually going to pour off the supernatant, the isopropanol. Again, I use my phone flashlight to keep an eye on the DNA pellet while I pour it off. Basically, when you pour it off, you really want to make sure you don't lose the pellet. So I use my phone flashlight so I can watch it as I'm pouring off the supernatant, the liquid. And it's okay if you don't get it all out. The more important thing is to make sure you don't lose the pellet. After the first 10 minutes, after the 10 minutes of centrifuging, the pellet should be pretty much stuck to it, so it isn't as much of a concern when you're doing the isopropanol. But after you add the ethanol, then it's going to be a bit more of an issue. So the next step is adding 900 microliters of ethanol, of the 70% ethanol specifically. So again, when you have more than one sample, you're going to want to make sure that you just hold it above it instead of putting it in there, because otherwise you'll have to reuse pipette tips. And then you're just gonna invert it a couple of times in order to mix it. Make sure the DNA pellet is washed. And then this is the really fun bit, because this is when the DNA pellet is gonna try to escape. When you pour off the ethanol, the DNA pellet is gonna try to escape. I figure out where it is first. Right now it's at the bottom. And then I just basically stare it down with my flashlight as I'm pouring off the ethanol. And the second it looks like it's gonna try to go for it, like make a run for it, I'm just like, nope. After that, you're gonna add another 900 microliters of ethanol. Again, holding it above so you don't have to reuse pipette tips. Mix it back and forth again. And now it's ready for centrifuging for another 5 minutes. So the next step is just centrifuging again for 5 minutes. Again, you twist until you feel the resistance. Close. Make sure it's the highest speed. And start. Yeah, so once you're done centrifuging it for another 5 minutes, you just pour off the 70% ethanol again. Like before, you want to kind of just keep an eye on the little guy. Sometimes he's stuck to the wall again and it's pretty easy, and sometimes he's still like free floating and you have to be careful. This time he was stuck to the wall, so it was pretty neat. These are going to be centrifuged again for another 30 seconds. So then we're just going to centrifuge for another 30 seconds. I usually just stand here and wait because like there's literally no point in actually trying to go sit down. So again, tighten firmly, set it down to 30 seconds, then just press start. Yeah. So last step before you put it into the incubator is you're going to want to pipette out any remaining liquid. So I usually use the P200 for this, and what I do is I have the phone camera, the flashlight set up. I reach in and I just carefully get the liquid out from around the DNA pellet. You want to try to grab as much liquid as you can, and then just it goes in there. Yep. So right now it's at 36.4. It's fine as long as it's like close to 37, then it doesn't really matter. what you're gonna do is you're going to make sure that all of the lids are like fully open on your samples, and then you're going to put them in the incubator, like sort of towards the middle, and then they're gonna be there for about 1.5 hours. It's okay if it goes a little bit over, but you want it to be in there at least 1 hour and 15 minutes, and then you don't want to leave them in there overnight, or else the DNA will dry out and become brittle. So once you have them all dried out, the last step you're gonna do, you're gonna take the P200 again, put it to 50 microliters, and then you're going to add 50 microliters of double distilled water to each of the tubes before going over to here. This is the negative 20 degrees Celsius upright freezer. You're gonna go in here, take whichever of these boxes that they go into. For us, it's gonna be in here, and then you're gonna put them in.